Welcome to the AKCI KCA lecture series. We're pleased to have today Gabriel Malouf, who's been a leader in kidney cancer for many years, I think mostly in the rare kidney tumor arena and also in sort of the molecular biomarker arena where we have a lot of shared interests. And Gabriel, we really appreciate your time and expertise in joining us today and look forward to hearing about biomarkers of response to treatment.
Yeah, thank you so much, Brian, for the kind invitation. It's a pleasure to be with you today. So I'm, for those that do not know me, I'm physician scientist working in Strasbourg University Hospital. So I have a clinical interest, obviously, with rare kidney cancer. And we've been developing some biomarkers of response to treatment. So today, my presentation will be in three parts. The first part is related essentially to rare kidney cancer and the effort we are doing in France to work on these rare tumors. So we have a network named CARAR network, so I'll talk to you about first. Then I will tell you two stories that we have published in the last 3 years about the mechanism of development of renal medullary carcinoma and MIT family translocation renal cell carcinoma. And finally, I will finish with what we have done essentially using bionic samples to predict response to immunotherapy and what we have developed in terms of biomarkers. And essentially, I've been focusing on epigenetic biomarkers of response to treatment. So to begin, first, I'd like to say that the kidney is a very complex organ. It's composed essentially from the tubules. Tubules have different parts, proximal part, loop of Henle, thin descending limb, thin ascending limb, and finally you have the collecting duct. And from each of those cells, you will have a different type of kidney cancer with different biology, different genetics, different epigenetics, different tumor microenvironment, different outcome. And therefore, it's really very important to try to link, and what we have been doing in the past year, to try to link each type of kidney cancer to the cell of origin and try to understand what is specific to this type of kidney cancer. So today we have 21 types of RCC. So obviously ccRCC is the most common type of kidney cancer. And then you will have a papillary RCC, but there are many new molecularly defined entities. So I think in this community, a lot of you are familiar about the TFE3 rare range tumor, TFEB altered tumor, FH, SDHB, ALK, and so on and so on. And obviously, little is known about these tumors. So in France, the National Cancer Institute tried to make the management of those patients with different type of kidney cancer even across all regions in France, and their focus is to decrease disparity in the management of these, of all type of rare cancers. So the French National Cancer Institute has established networks covering all France. And among these networks, we have a network that has been recently accredited by the National Cancer Institute, focusing on rare kidney cancer named CARAR network. So this network that I'm coordinating have 9 missions. First mission is to give pathological and clinical expertise for the community, allow the patient to access to specialized center, inform patient and family, optimize patient pathway, contribute to best practice guidelines, education of professional, promoting clinical trials, and have epidemiological surveillance and data management. So in France, we have 14 expert centers. In each center, you have pathologist, medical oncologist, and surgeons. And the goal of these 14 expert centers in each of their region is to ensure that the management of these patients is very well done, that we can review all the pathology slides if needed, that we can do all the molecular testing when it's necessary, and of course, collect the clinical data and do research on those samples. So, as I said, we have 15 expert pathologist colleagues across all France that review annually 1,200 cases, 600 cases per year. They confirm that they are rare kidney cancer, and they meet every month where they have around 10 cases that are complex cases where they can discuss all together about them. They perform molecular testing in all papillary RCC to ensure that it's not FH, SDHB, SMART-B1, TFE3, TFEB. And at the end of each pathology report, they indicate that the case has to be discussed in expert centers and essentially in a national multidisciplinary tumor board. So this is the first thing that we do in the network, the pathology review. The second thing is we have established an online MTD. So since the 3 years now, we have online MTD, and in this online MTD that's national, we have 2 pathologists, 2 medical oncologists, and 2 urologists. So we meet every 15 days, and we have 20 cases that are discussed coming from all France with different type of pathologies. The number of cases has been increasing. So since 2023, we begin in February 2023, we discussed over 1,000 cases so far. Essentially, the cases are metastatic case. We have some cases with locally advanced tumors and localized, but they are very rare. So median patient age around 61 years. We give, the response is very quickly. Median number of days between the time where the doctors submit their cases and the response is around basically 5 days. And essentially, the majority of the patients that has been discussed, as I said, is metastatic tumors with intermediate and poor risk disease. And here are the data regarding the tumors that has been discussed in our network. So it's a little bit old numbers, but the statistics still hold. What you can see is we have only 32% of papillary RCC, followed by chromophobe RCC is 21%, and then you will have all the rare entities coming afterwards with TFE3 translocation, which is around 10%, SMART1 deficient tumor, TFEB, FH, oncocytic tumors. So we do not have really the same statistics as you can see with the big database like IMDC, where you see papillary 55%, chromo 15%, and unclassified. In our cases, the majority of the cases has been classified, and we have really an accurate, I think, job that our colleagues pathologists are doing to cover all the nation and to provide, accurate diagnosis, and they can reduce the disparities across regions. This is the first point that I want to discuss with you. The second area of interest to my team since now, I would say 15 years, since I was a fellow in MD Anderson back to 2010, 2011 with Dr. Tania, is to try to better understand rare kidney cancers, better define for these rare kidney cancer the genomic alterations, what's the mechanisms that's involved in their transformation. I'll not have the time to go over all what we have done in the past 15 years, but I will just focus on 2 stories. First one is related to renal medullary carcinoma, and the second one is related to MIT family translocation renal cell carcinoma. So renal medullary carcinoma is very rare kidney cancer that arise in children and adolescents. It's characterized really by SMART-B1 loss, which is the feature of those tumors, and it arises exclusively in patients with sickle cell trait, sickle cell disease. And there has been really few studies that investigated the molecular underpinnings of these tumors. So the first effort that we published in 2020 was Pablo Sumsaul and MD Anderson. So this began working on this in 2010. It ended up being published in 2020. So we collected at that time the largest cohort of renal medullary carcinoma, and we did a lot of testing, including exome sequencing, RNA sequencing, and DNA methylation. And we tried to describe in this effort, the patterns of SMARCB1 alterations, and 50% were related to one mutation and deletion of the second allele, and the other one were related to translocation, basically that leads to loss of function of one allele, and we have also LOH of the second allele. And what we have shown that renal medullary carcinoma, this is RNA sequencing data, and this is the convolution of immune cells using MCP counters, that as compared to other type of kidney cancer, essentially chromophobe, RCC, that these tumors are highly inflamed, and you have a lot of fibroblasts that are associated with these types of tumor. But we do not know at the single cell level how the ecosystem of renal medullary carcinoma is organized. And here we have done the first single cell RNA sequencing of renal medullary carcinoma. And we did along the single cell analysis of the primary tumors, single cell analysis of the normal nephron. And what we have shown, and I can show you here in the slides, sorry, it's very busy. So this is basically proximal. This is UMAP showing basically the different cells associated with normal kidney. So you see here the PCT, proximal tubule, that is here. And then you have the TAL, the thin ascending limb, the distal convoluted tubules, the connecting tubule, and the distal tubules. And here what you can see is the RMC cells. So basically RMC cells are located close to TAL, thick ascending limb, and we showed that we have a high correlation between the gene expression signature of RMC cells and TAL, suggesting that the thick ascending limb TAL is the cell of origin of RMC. We have shown that these tumors are highly infiltrated with a lot of cancer-associated fibroblasts. So you see here that we have a lot of fibroblasts. And we show that as compared to the normal cell of origin, the TAL, they lose basically OXPHOS and adipogenesis, and the tumors were highly enriched for EMT, inflammatory signaling, and hypoxia. We also have done, the analysis of the cancer ecosystem in 3 tumors, and we showed that often RMC, as you see here, in the ecosystem, there's very few T cells, and there are rather plenty of cancer-associated fibroblasts, as you can see here, and tumor-associated macrophage that are really composing the cancer ecosystem. And we also have done like pseudo-time analysis to better dissect the plasticity of these cancer cells at the single cell level, and we show that basically they can be divided in different population. You have a population that is epithelial-like, as you see here, that's overexpressing EpCAM, PAX2, CDH1. And this population is evolving toward a population that's fully mesenchymal, as you see here, that expressed fibronectin and CDH2. And you have another population that is rather mitotic, as you see here, that's highly proliferating. So essentially, you have an epithelial population, you have a mesenchymal population, and you have a population that is really mitotic and highly proliferating. And then we analyzed 4 RMC cell lines that are currently available, and we find that the RMC cell line can recapitulate human samples. So we have one population, one cell line that's RMC219, sorry, that's essentially highly epithelial, as you can see here. highly epithelial. And you have RMC2C that is here. You see, it's highly mesenchymal when we have done FACS sorting of two markers, EpCAM, that is epithelial marker, and CD44, that's mesenchymal marker. So it's a spectrum. RMC219 is epithelial, and RMC2C is highly mesenchymal. And then you have UK353, that's mesenchymal, as you can see here, and the UK360, that is hybrid. So it has both epithelial and mesenchymal. And the most aggressive line is, of course, the mesenchymal one, because using Matrigel and to test the migration, we showed that the RMC2C is the most invasive, cell line. So those cell lines basically recapitulate what we can see in the single cell data. And as I said, these tumors have been highly infiltrated and they are associated with the presence of different type of CAF. I would not have the time to go over them. And essentially, you have two type of CAF. You have the myofibroblastic CAF and you have the ICAF. And we have shown that in the metastasis, you have increase of the presence of these types of CAF. Sorry, here the slide is a little bit blurry. So doing the single cell sequencing has been really interesting, as I said, because it allows us to see that the RMC cells are very close in their transcriptomic program to the thick ascending limb. Then we tried to look in single cell RNA sequencing to transcription factor, basically, that are overexpressed in RMC cells and that are lost in TAL, and the opposite transcription factor that are activated in the TAL and are lost in the RMC. And what we have found is that you have several transcription factors that are lineage-specific transcription factor defining the TAL, such as TFCP2L1, MITF, OXB9, ESRA. So those transcription factors were expressed in TAL and are lost in the tumor. And you have other type of oncogenic transcription factor, basically, that are not present in TAL, but they get expressed in RMC. So you have a kind of balance. When you have cancer, you have to lose the lineage-specific transcription factor, and you acquire oncogene-specific transcription factor, and we've been able to show in two different cell lines. So here you have the RMC2C cell line, that's mesenchymal. So this cell line is with mCherry, and this is the RMC2C when we try to overexpress SMARCB1. So this is the mesenchymal one. And we have the same thing in the epithelial one. So without SMARCB1, so we have mCherry, and with SMARCB1. So what we've been able to show that following expression of SMARCB1, you will have increase of the expression of lineage-specific transcription factors, so TFCP2L1, MITF, and you have decrease of oncogenic, transcription factor that are really decreasing. And what we have always also observed, and I will show you the data later on, that basically upon expression of SMARCB1, the cells will die by ferroptosis. So the ferroptosis is a mechanism of cell death that's associated with iron overload. And we've been showing that SMARCB1 basically is regulating the ferroptotic resistance. And this is IF.
Can I ask a question about that?
Sure.
So it looks like when you have SMARCB1 added back into the cells, you're not only getting expression of that protein, but you're essentially stabilizing several other switch-SNF complex proteins. So probably the entire complex is either there or falling apart in the SMARCB deficient cell. Any thoughts on the biology of how that might be impact? Like, in other words, what is the selection pressure to lose SMARCB1 and not like some of the other members? You would imagine that you would have redundant outcomes perhaps if you did lost any of the other complex members as well.
Yeah, so this is a good question. So here I showed you just few genes, members of the complex. So we see some changes in basically DPF3 that's coming back. SMARCA2 basically that's not here and is coming back again. And now we have a postdoc in the team that try basically to understand better, the complex and what's going on there. So we're doing mass spec to try to understand basically what's going on for BRG1, where it's really binding. So we don't have the data yet, but we're working on it. can proceed. So what we have shown is that that transferrin receptor basically, which is the receptors that allow the iron to enter the cells. So following SMARCB1 reintroduction in the cell, so you will see that you have increase of the TFRC. So you have iron overload, and as you can see here, the cell will die quickly following SMARCB1 reintroduction. And basically, we have done cell viability of different cell lines. We treat them by RSL3, GPX4 inhibitors, and we showed that the RMC cells are highly sensitive to different agents, basically, that activate ferroptosis. And if we use ferrostatin, we've been able to stop the process, and it's not the case by using camptothecin. I will not go, deep on this. And then we try to look basically at the complex, and to see we did the ChIP-seq for SMARCB1, BRG1, HTK27 acetylation, and some AKCI. And what we have shown is that following introduction of SMARCB1, it will retarget the SWI/SNF complex to enhancers of sick ascending limb cell identity. And you will find the complex basically in TCP2L1, HOXB9, MITF, and PAX8. The second rare disease that I would be talking on is MIT family translocation RCC. So it's one.
Gabriel, can I ask a question before you move on to this? I think what surprised me in one of your first slides was about the immune infiltration of medullary, which is obviously not immune responsive, and I think is hyperprogressive, right, on ipi/nivo.
Yes.
Is it just that they're exhausted T cells? Is it just that there's a big fibroblast infiltration, or, why aren't they doing anything, I guess, my question.
Yeah, I think, from our data and from what has been described in the literature before our work, the pathologists, when they look at RMC, they see that you have a very highly desmoplastic stroma. And in this desmoplastic stroma, you have a lot of fibroblasts. And when we have done our single cell data, we have shown that at least in the samples that we analyze, we have very few T cells. And the majority of the cells that we are getting from the microenvironment are either basically myofibroblastic calf or are macrophage type 2 macrophage. So I think this is one of the reasons where those tumors do not respond to immunotherapy, because you have a lot of myofibroblasts that act as a shield that do not allow basically immune cells to penetrate in the tumor. At least this can be one of the explanation. And second, you have a lot of, macrophage type 2. And we know that whenever you have a lot of macrophage type 2, you will not respond to immunotherapy. I think Pablo Sawel has doing a lot of job, in this setting, and that 2 trials stop accruing in MD Anderson with IO because of rapid progression under IO. But I think that the presence of MyCAF and the presence of M2 macrophage can explain this.
Yeah, great. Thank you.
So regarding translocation RCC, so I began, as I said, working on this almost 15 years ago with Bernard Escudier when I was fellow in Gustave Roussy. And our early work, we're trying to understand if these patients respond to TKI. So at that time, we collected data from different centers in France and US. And we find that the median PFS on TKI was around 8 months. And then we have collected the data of patients treated by IO or IO, and the response of treatment was very low. At the same time, while we've been collecting these data, we try to get the material to better understand the transcriptomic features of those tumors, to understand, the genomic aberrations of these tumors, and also to try to understand the mechanism of transformation of translocation RCC. So we published now 10 years ago that translocation RCC, if you compare them to other RCCs, they have really unique gene expression signature. They have no mutations of BHL, PBRM1, and BAP1. The data has been reproduced by other teams. But we find that one of the major determinants of aggressivity of these tumors is essentially presence of chromosome instability. And if the patients basically have a lot of chromosome instability and a lot of copy number gain and loss in addition to the translocation, and here we have published in 2014, the patient have in the tumor 17q gain, you see that they do poorly. And if they don't have 17q gain and they have a normal karyotype, basically they are doing well. Often pediatric patients have this profile. They have a profile with no genetic instability, while adult patients have really a lot of alteration and 17q gain. And this has been also validated by colleagues in Memorial. And we try to better understand basically what we published recently, the role of TFE3 and how TFE3 basically is leading to oncogenesis. And what we have done here is we have knocked down TFE3 in different RCC lines. And you can show if we knock it down, we decrease the colony formation in different translocation RCC line. But we didn't know what's the program related to TFE3. So we have done RNA sequencing in 4 cell lines. And gene set enrichment analysis has shown that in 4 cell lines consistently, whenever you knock down TFE3, you have decrease of oxidative phosphorylation, suggesting that TFE3 basically is regulating OXPHOS. And we have shown this as we have done seahorse analysis and shown change in the oxygen consumption rate in these types of tumor. And we have similar, results that has been obtained by Srinivas team in Dana-Farber. We have done the opposite experiment. We have done gain of function of TFE3 alone, mono-TFE3, PRCC TFE3 in HEC cells. We have shown that if you do the gain of function again, you obtain increase of oxidative phosphorylation, suggesting that again TFE3 is regulating OXPHOS. And we have done genome occupancy of TFE3. So we did cut and tag for TFE3. We did mapping of H3K27 acetylation, H3K4 thymethylation. So we obtained plenty of peak related to TFE3. Overall, in these 4 lines, we have 84,000 peak that are associated with TFE3. And when we look basically at the core binding of these TFE3, we looked at the promoters where it's binding. We find enrichment of lysosomal genes. This is what has been known before, autophagy, mitophagy. But we also have found that TFE3 is binding to genes related to ferroptosis. And again, we used RSL3 to basically inhibit the ferroptosis in these tumors, and we show that the TRCC cells are highly sensitive to ferroptosis inducer, and we can block that by adding ferrostatin. Something also that's important, I'll not have the time to go through it, is that if we analyze several patient cohort of TRCC, and we looked for the OXPHOS score, and here you can see this is some analysis in our single cell data for the OXPHOS score. And in CCRCC, you can see that the OXPHOS is decreasing with EMT, so the epithelial cells have high score of OXPHOS, the same thing as the proximal tubule. And then basically, as soon as they become mesenchymal, they shift toward more glycolytic activity. And this is why their OXPHOS score is decreasing. But we have shown that in translocation RCC, these cells, wherever they are epithelial or mesenchymal, they maintain a high score of OXPHOS. And obviously, you know, OXPHOS is now very important. There's a lot of papers that has been published in the last 2 years suggesting that OXPHOS is highly activated in metastatic CCRCC sample, and that the tumors that have high level of OXPHOS is associated with poor outcome. We have shown also that the tumors that have mesenchymal signature and MyCap again are associated with poor outcome. So this is the second point. And finally, I will finish by the third point that I would like to show you today is what we've been doing in the biomarker field, and essentially analyzing bionic cohort regarding the response to immunotherapy. So before going to discuss specifically epigenetics, all of you know that CCRCC is a disease that is poorly, basically we have low mutation load. And despite the fact that CCRCC have very low mutational load, CCRCC respond very well to immune checkpoint inhibitors. So therefore, we do not think that only genetic can explain, the response to immunotherapy. So with a low mutational burden, it's not genetics that will explain who are the patients that are responders and who are the patients that are resistant. So we thought in our team that there might be different layers that can basically dictate the response of patients to immunotherapy. One of them is epigenetic program. The second could be the activation of ERV and viral mimicry. The third is cell plasticity, and finally the changes in myeloid vascular escape axis. So in the time that remains for me, I would like to discuss essentially what we have found regarding epigenetic profiling of bionic samples, and I will present some data regarding cell plasticity. So when we think about epigenetics, different from genetic, where you have a single base or many bases that are mutated with known, effect. Epigenetic alteration, essentially DNA methylation, can occur in different regions of the genome. They can occur in promoters. So if you methylate promoters in CPG island, it might lead to inactivation of genes, but also you can have epigenetic alterations occurring in enhancers, and obviously the effect is not the same. And you can also, as you know, whenever we have cancer, we have methylation of promoter CPG island, but we also have global demethylation, and when you demethylate globally the genome, you will activate a lot of repetitive element, line, sign element, and a lot of ERV. So I will show you some data where we integrated the analysis of DNA methylation, transcriptomic, genomic, and clinical data. And I will show you what we have observed. So the first thing that we discovered is that we discovered that by analyzing 5 cohorts, so 4 clinical cohorts and 1 CCLE clear cell RCC cell line cohort, that you have a subset of tumors that have enhancer demethylation. So you have a subset of clear cell RCC that have demethylation of their enhancer. So the demethylation of enhancer regions is often present in hypomethylated tumor. And as you can see here, you have a group here that is demethylated, okay, that we call TED, and the tumors that have enhancer methylation are often in different cohorts associated with poor outcome. We have also shown that whenever you have this phenotype, the TED, it's often associated with expression of TET1, and basically the promoter of TET1 is hypomethylated and associated with TET overexpression, and this might explain why these tumors are really hypomethylated in their enhancers. And then we look by the convolution to check what is the tumor microenvironment landscape of these TET positive tumors as compared to the TET negative tumors. We find that in these TET positive tumors, we have high expression of immune checkpoint, and we have infiltration with both active and suppressive population. And these tumors that are TET positive are highly enriched for the presence of CAF. They are highly enriched for 9p loss, and they lack often the mutation of PBRM1. And in contrast, the TED negative group is essentially enriched for the presence of endothelial cells. And when we apply this signature to bionic cohort, we have two clusters. We have this cluster of tumors that are TED positive, and this is a cluster of tumors that are TED negative. So obviously, the number of samples is not huge, but you can see that basically the TED positive tumor are enriched for TNF-kappa B, they are enriched for EMT, they are enriched for interferon signaling. The patients that are TED positive progressed quickly on Ipi/Nivo, and they were associated with poor overall survival as compared to the patients that are TED negative, indicating or suggesting that epigenetic architecture is important for predicting a response to immunotherapy. So this is the first analysis that we have done, and been really focusing on enhancers. So then we decided to focus more specifically on promoter CpG island, because it has been shown that whenever we have promoter CpG island methylation and we have repression, these might be the genes that are really important with functional relevance. And we have done, the same pipeline as the paper on the PED and TED, and we analyzed 4 different cohorts. We integrated the DNA methylation and expression, and we have found that you have a group of tumor that consistently basically methylate a subset of gene and repress them. And this group of tumor, we called it IMS high, for index of methylation high. And you can see that these tumors have poor outcome in TCGA and different others cohort 2. And we have identified basically consistently in different cohorts a signature of 21 genes that are methylated and repressed, and we have applied it to bionic cohort. And as you can appreciate from the Kaplan-Meier curve, you can see that the tumors that have methylation and repression have poor outcome as compared to the other one. They have also 9p loss and poor outcome. And then we have created basically an index for DNA methylation that you can compute, and you can check if the tumors can be sensitive or resistant to IO. I will go quickly because we tested the score against other established signature, and we find that it's independent of other signature. And what is really important, and this is we discussed for Brian, for many times, what we have found is that in TCGA, in bionic cohort, if the index of methylation is really high, you have more immune infiltration. But in the tumors that have low IMS, so have less basically methylation, that they respond to IO, they are associated with the presence of basically endothelial cells. Again, IMS is prognostic, but is also predictive of response to immunotherapy. So you see if the tumor have high IMS score is basically poorly responsive to IO, but it's not the case in bionic in the TKI arm. And we have used another study with 102 samples treated by TKI, and we didn't find basically differences or significance regarding the response to TKI. So we can say that the IMS score, the prognostic factor that tightly associates with patient outcome, but is also a predictive indicator of resistance to immunotherapy. And therefore, we can divide basically the tumors in Bionic in two groups. You have this group below, where we have silencing of genes at transcript level by DNA methylation. It has poor outcome. It has high immune infiltration. It has endothelial depletion, immune evasion, EZH2 activation. And you have signature of BAP1 and SETD2 deficiency. And you have the other group here that have low basically methylation or no hypermethylation of these promoter that's often associated with good clinical outcome, low immune infiltration, and high endothelial component. And this group is sensitive to Nivo or Nivo-EP in Bionic. So based on these data that we observed, even though the number of samples is really limited, we have just 49 samples, and it's very challenging to do. So we try to come up with a way just to use the iMotion 151 signature. So of course we didn't, we use kind of Z-score to try to put the samples in C1, C2, and C4, C5. C1, C2 were the angiogenic basically signatures, C4 and C5 is immune signature, and already in Bionic in the publication where they try to allocate the treatment according to 1, 2, 3, 4 signature, the 2 signature of Bionic is associated with angiogenesis, and already in Bionic what we can see, if the patients were allocated to the Ipi-Nivo arm, you have 51% response to IO and 50% response to VEGF or TKI. And what we have found if we use, the iMotion signature is that the patients that clusters with 1 and 2 have a response rate of 70%, and the patients that cluster to 4 and 5 have 41% response rate, and the complete response are all in cluster 1 and cluster 2. What I would like also to highlight that in this cohort we have a lot of favorable risk patient, and you can see that in C1, C2 we have a lot of patients that are favorable risk. So maybe the results can be different if we use population with intermediate and poor risk only. I will not have the time to go in what we have done in cell plasticity to let you ask maybe a few questions, but I can maybe just finish with one thing is we have done some single cell sequencing on ccRCC, and we find that basically the tumors can be divided in 4 cell state. You have the epithelial state, you have the intermediate state, and you have here the mesenchymal state, and you have an inflamed state. The inflamed state is a state where the cells are really mesenchymal, but they express a lot of MHC class 2. And this can be associated with the presentation of the antigen to CD4, and it can play a role in response to immunotherapy. And what we have found in Bionic is that the mesenchymal cells, by the convolution, associate with MyCAF often, and that the mesenchymal cells, as you see here from spatial data, associate with MyCAF, often at the border of the tumor. So here you have the tumor border. This is the tumor, and this is the periphery, and the MyCAF and the mesenchymal cells go together. And what we have shown is that in Bionic, whenever you have high level of MyCAF, it's associated with poor progression-free survival and poor overall survival. And I think it's very important to target these population of MyCAF. And in our manuscript, we have identified a lot of ligand receptors that can allow the MyCAF to interact with mesenchymal cells. So just I'll finish with this slides. Say rare kidney cancer are not rare. They present 25% of all cancers. Often they are aggressive. So there's a need really to work on these types of tumor, and we need collaboration between different centers in the world to push the needle forward. SMARCB1 and TFE3 regulate apoptosis-resistant programs that might explain their development. TFE3 is regulating oxidative phosphorylation, indicating the importance of metabolism in rare kidney cancer. Epigenetic programs define the biological states that shape sensitivity or resistance to immune checkpoint blockade in CCRCC. And I think the future is to integrate epigenetic architecture, viral mimicry, spatial tumor ecology to come up with a better prediction to respond to immunotherapy. And I would like to thank all my team at IGBMC that been working for these last two stories. Thank you very much. I'm looking forward to take any question.
Gabriel, thank you very much. That was a great, amazing talk. Congrats on your, body of work over the last 15 years, really across rare kidney cancer and the molecular. I guess I'll start out with a question, and just so the group knows, Gabriel and I have had extensive offline discussions about this finding that the angiogenic clusters and the response to immune therapy, there's some methodology issues, et cetera, and we're meeting monthly to sort this out. So I don't want to dive into that rabbit hole. I guess I'll buy that the angiogenic phenotype has also an immune component, right? And that's good news for IO-TKI, right? Because we don't really give TKI monotherapy, at least in frontline. But I think if I got the slide right, you also showed that it wasn't really associated with outcome to angiogenesis inhibitors, right? From the other cohort.
Yes, exactly.
So I guess that's the part that I don't get, right? If these are truly angiogenic tumors, we should at least see something we should see some differential effect to TKI monotherapy. To me, that stands to reason, but maybe I'm not understanding it.
No, we didn't, the data that we looked for, it was for TKI, we have a cohort of patients treated with sunitinib. So these are all TKI, and the mechanism of action are different from new TKI, because if you take cabozantinib, if you take lenvatinib, we act more on, AKCI, MET, FGFR. And as I showed in my slides, I think you have two different types of tumors. You have the tumors that are basically resistant to IO have EMT, and this has been shown by others. Yeah. And whenever you have EMT, you have activation of AKCI, MET, FGFR. So this is why I think, maybe next generation TKI might work in this group. Essentially is not through angiogenesis, but it's through targeting EMT, essentially. But I mean, this is hypothesis generating. I think it's essentially through this, but of course it might act through angiogenesis. So I think when we say angiogenesis is vague because, as you know, the cabozantinib and the lenvatinib are kind of dirty drugs. They target VEGF, but they target something also else. So they might also acting not only on the vessel, but they might also act on cancer cells. So this is my interpretation.
Yeah. I mean, again, I don't want to dive too far down the rabbit hole. We can talk about it later, but I think when we say angiogenesis, right, it's not one thing, right? It probably means different things. So, I'm thinking of it very simply that for sunitinib or pazopanib or axitinib or anything, if you have an angiogenesis signature, it should be associated with outcome to those, certainly the more pure angiogenic agents. For the less pure ones, there's probably other biology and other targets. I totally agree. So anyway, we'll chat more about it. I'm still trying to understand it.
Sure.
Other questions from the group?
Yeah, I have a question. Hi.
Kelly.
Good morning, Kelly. I'm GU Med-Onc at UCSF. Nice to meet you. Thanks for the talk. I'm curious about the IMS high clear cells and whether there are manifestations of that we might be seeing already in our clinical observation. So I guess one question I have is, do we know if there's any histologic feature, maybe sarcomatoid or otherwise, that are more frequent amongst this IMS high population? And then I'm also curious, what is the degree of overlap between IMS high status and VHL loss that's driven by promoter methylation as opposed to coding sequence mutation or deletion?
Yeah, I mean, these are good questions. Whenever you have IMS high, it's essentially associated with aggressive tumors, essentially. So more methylation you have, the tumors are associated with higher grade tumors, essentially. So regarding VHL methylation, we didn't check. VHL methylation is not common. It's like 6% of the ccRCC have VHL methylation. And VHL methylation didn't come up among the genes that were differentially methylated between IMS high and IMS low. So if you check those genes, we have a set only of 21 genes that were recurrently, methylated and repressed in different cohort. I know DNA methylation is different, I think, from mutation because if you activate EZH2, you might, lead to a global effect. So I don't know if the effect is really coming from specifically the methylation of those genes or from a phenotype that's generated by activation of EZH2 or the NMTs and leading to, what we see.
Yeah, I would be interested to know. I don't know a whole lot about this less common subset of clear cells that have VHL loss because of promoter methylation, but it would just be interesting if that was a byproduct of this phenomenon.
No, it's not a byproduct. No, it's not byproduct of this phenomenon because, to my knowledge, I don't know if there's a lot of cohort investigating, if the tumors with VHL methylation behave differently than the tumors with VHL mutations. This is we have not investigated so far from our side, but I can tell you it's not related to IMS. Otherwise, we would see it there, but it's not there.
Thank you.
Thanks.
A couple of points, I think. So one, directly in response to the previous question, I think the one thing that we are not talking about in this conversation is the fact that in VHL deficiency or even in tumors with VHL intact with physiological hypoxia, activation of HIF is going to also drive up the expression of many of the demethylases. In fact, the KDM6A, KDM5C, both of them well known to be mutated in both in the TCGA cohort, but also we routinely see inactivation of these particular demethylases, and this is going to impact the methylation profile of your tumors, right?
Yes.
In the same light, the DNA demethylase, the TETs, for example, I think TET3 oftentimes we end up scoring. We don't know how much the expression is and what the functionality is, but the TET is another enzyme that should be a part of these conversations. So I think there's enough literature to suggest that there is dysregulation of methylation sticks in RCC, but it's also fair to say that mechanistically we do not understand sufficiently why this is occurring, and it is unlikely to be just one enzyme or one truncal feature that is driving it. It's more likely to be a combinatorial effect of many converging signals coming onto chromatin in this case. So I think it's an important conversation, but I think it's not a very simple answer either. So we need to keep thinking of that. The second part that I actually wanted to ask, and this from a general community perspective, with all of these cohorts coming in, are we trying to create easily accessible interfaces where, for example, we could start using these patient data sets and like start plug and play with regards to our genes of interest? In many instances, wherever a skillful computational biologist has gone and done that part, discovery has been expedited pretty remarkably, I would say, because each one of us has our own biases, and so we keep looking at the data with our own blinders on. But it's probably going to be a much larger and useful enterprise if we can think along the lines of the AKCI, Brian, to start making this usable in a general, like create a website where you can just say, okay, this cohort, this gene, show me what the outcome, what the expression levels, where does it go what are the correlates? It would be a game changer, I feel.
I would agree with you. Yes, I agree with you. I could not agree more. I think this is very, very important. So obviously, in all the trials, as you know, that has been published, you have to rely, on not only the PI, but who's the promoter of the trial, right? So patient consent, all, the requirements that can allow these data to, obviously, as you said, to belong to the community, that everybody can check exactly their gene of interest, their pathway of interest. And I think we've been fortunate at least to work on, epigenetic for bionic trial, but I know that for a lot of trials that are industry sponsored or others, I mean, we have very few data available on the role of epigenetic in this setting. So I'd love to see, data coming in this field to see if what we observe is basically correct, it can be optimized. But so far, I think the field is lacking data regarding epigenetic. We see a lot of data regarding transcriptomic mutation, but there's no data on epigenome. And I think we should go maybe beyond DNA methylation by investigating, histone acetylation or histone methylation, but there's lack of data already in this field, and I think we need, as a community, to try to see. So we tried in several sponsored trials to ask the companies if we can share these data, but we never succeeded to do that.
Yeah, Abhishek, Gabriel and I have talked about amalgamating some data, but it's challenging, right? There's sponsor issues and and they won't release the clinical data. Some of it's public, some of it's not. So it's very challenging, but I think your point's well taken. Let's do Stephen for one more quick one before we go.
Yeah, I just have a quick one. I was wondering about these like epigenetically driven aggressive tumors, I guess the IMES tumors. Do you have a feel for where they develop in RCC evolution? Are they like a early event that gets fixed, kind of VHL loss, PBRM mutation, then this occurs, or is it something that occurs later down the road, like as we're approaching metastasis, once whole genome instability index is going high and things like that?
This is really a great point that you raised. I think so far it's really a speculation. So what is sure is that like you have some type of tumors like FH deficient tumor, SDHB, it's known that really they arise very early, because whenever you do mutation of FH or SDHB, you will have hypermethylation right from the beginning. But how it's going in CCRCC, my interpretation is that it's a later event, probably. Why? Because when you looked like IMS data, we show that the high IMS is associated with 9p loss and is associated often with SETD2 mutation. As you know, the 9p loss occurs late. SETD2 also you have a parallel evolution. It's not early event, so it occurs late. So it's highly possible, I think, that as tumors evolve, they change their epigenetic machinery as they drift by losing 9p, and then with the division they accumulate DNA methylation. But we do not have data to show. So beyond the association with 9p and SETD2, we do not have further data. I think we should definitely check that. The issue with this is it's complicated. I think nowadays, even though we have a lot of progress with single-cell RNA-seq and ATAC-seq, but there's no data in single-cell DNA methylation that's published in CCRCC. But it's something that's it's worth investigating and to see how this DNA methylation basically is changing during EMT and during the loss of 9p.
Yeah, it seems like matched primary compared to MET, which obviously there isn't a lot of methylation data on, would be one way to look at it. That answers my questions, though. I appreciate it.
Thank you.
Thanks, Gabriel. Really appreciate your time and expertise. Thanks for joining today. And
thank you for the
thanks
invitation.
Everyone for joining. I think we have a quarterly meeting coming up in a month. I'll be in touch with some details. So thanks all. Have a great day.
Thank you.
Thank you all. Bye-bye.